The protective effects of liguzinediol and congestive heart failure induced by myocardial infarction and its relative mechanism. This is presented by Valentina Ramirez and Andrea Ramirez.
Congestive heart failure. It occurs when the heart does not pump blood as well as should. Some causes may be narrow arteries in the heart, high blood pressure, diabetes, or congenital heart defect, which leave the heart too weak or stiff to contract and pump blood. Some symptoms may be dizziness, fatigue, weakness, aching in the legs, ankle, the feet, couch, or wasting ascites.
Liguzinediol is a compound synthesized after the structural modification of leukotriene, that is one active ingredient of Sichuan lavage rhizome. Liguzinediol could significantly exchange left ventricular contractility and improve the diastolic function of a rat heart without arrhythmia and other adverse effects. But it is still unknown whether liguzinediol can improve heart failure caused by primary myocardial damage, as transforming growth factor beta-1 TGF-beta-1 SMAD pathway plays an essential role in the pathogenesis of heart failure to detect the effects of liguzinediol on TGF-beta-1 SMAD pathway.
The general objective of this article is to share the effect of liguzinediol in the prevention of heart failure and to explore the related mechanisms.
The ELISA is a test that uses an enzymatic marker. The most used enzymes are radish peroxidase or alkaline phosphatase. These enzymes are used because they are cheap and pure, and normally they are not in body fluids, since if they were present could generate false positive or false negatives. For this test, we need a substrate so that the enzyme can fulfill its function. This type of tests are heterogeneous since we need washing during the process. The ELISA test has three types. So in the first type, that is called sandwich type, this type of test helps us to identify antigens. When there was a reaction, it is because the patient is positive for the antigen we were trying to confirm. Competitive type, in this type of ELISA, we seek to identify antigens. If I can see a reaction in this test, is because the patient will be negative for the antigen. And the third type, that is called indirect type, unlike the others, in this one we try to find antibodies. In this test, we are going to add an anti-antibody. If there is a reaction, it confirms that the patient has no immunological memory and it is positive. On the other hand, if there is no reaction, it is because the patient is negative and had no immunological memory. There was some blood, the expressions of proteins involved in the TGF-beta-1 SMAD pathway were detected. This is a technique used to detect a specific protein in a tissue or blood sample. Involves the use of gel electrophoresis to separate the proteins from the sample. Once separate, they are transferred to the membrane where they will be exposed to a specific antibody against the protein under study. Binding to the antibody is detected by a chemical or radioactive marker.
PCR polymerase chain reaction. It is an enzymatic amplification technique based on three reactions of repeated cycle, but varies the temperature. 1. DNA is the nature to separate the two strains. 2. Hybridization of single strain with an oligonucleotide or primer. 3. The single strain is replicated by DNA polymerase from the oligonucleotide. In the radioimmunoassay, the immunoassay is labeled with a reactive compound. The labels can be beta or gamma radiation. They can be equilibrium assays where the labeled and unlabeled antigen compete for the antibody to equilibrium, and the sequential assay where the unlabeled antigen is exposed to the antibody for saturation. The red immunoassay was used to detect inflammation and oxidative stress, measuring angiotensin, aldosterone, and plasma renin activity.
The weights of the whole heart and left ventricle were recorded. The heart mass index (HMI) and left ventricular mass index were also calculated. As shown in the table 4, both HMI and LVMI were significantly increased compared to the sham group, and left ventricular hypertrophy was observed in MI rats. All treatments could effectively reduce HMI and LVMI, while there was no significant difference between the MI group and the liguzinediol group. This is the
With the meson stain, we wanted to observe the behavior of the collagen fibers in the left ventricle. In MI mice, collagen was increased and necrotic cardiomyocytes were increased. Captopril, digoxin, and liguzinediol can reduce collagen depositions and myocardial fibrosis. The increased expression of matrix metalloproteinases MMP2, MMP9, and TIMP1 in heart failure and myocardial infarction were related to the deterioration of systolic function and the expansion of the ventricular cavity. Medication regulates the balance of MMP and TIMP. TGF-beta1 is the central regulator of ventricular remodeling, which participates in myocardial fibrosis process by regulating myocardial growth, myofibroblast activation, and the generation of extracellular matrix. The Western blot result showed that the expression of TGF-beta1, pSMAD2, pSMAD3, and CD105 were dramatically activated in my rats, and increases were reserved by captopril, digoxin, and liguzinediol.
discussions. Congenese heart failure is mainly manifested in progressive decrease of cardiac pumping function and remodeling of the ventricular cavity. Yes, agree. Q-T says that renin-angiotensin-aldosterone is a central system that regulates blood vessel tension, blood volume, and cardiovascular function. Renin-angiotensin-aldosterone also plays an indispensable part in this pathogenesis of heart failure. Yes, agree. Fu GH says that ligation of coronary arteries is a commonly used method to establish a rat model of heart failure caused by MI. And yes, agree.
In conclusion, we have that molecular biology helps to verify that the results after treatment are as expected, determining the composition and structure in a specific tissue of the organism in general. Molecularly, knowledge of living beings allows interaction between different species to find better treatment for various diseases. When knowing molecularly the process of acute myocardial infarction and heart failure, various pharmacological treatments such as liguzinediol, which is discussed in the article, can be present.